The Stanford Fermented Food study did not prove fermented food beats fiber for inflammation. Its own primary outcome was null in both groups. That sounds like a technicality. It is not. It is the difference between a study finding something interesting and a headline claiming something the study never tested. In 2021, Wastyk and colleagues published a widely shared paper in Cell called Gut Microbiota-Targeted Diets Modulate Human Immune Status. It compared two active diets, a high fermented food diet and a high fiber diet. The paper contained a striking result. 19 of 93 measured inflammatory proteins decreased in the fermented food arm. None of those 19 moved in the fiber arm. But the correct sentence is not fermented food beat fiber. The correct sentence is, within the fermented food arm, 19 proteins decreased, while those same within-arm changes were not observed in the fiber arm. The researchers did not report a direct between-arm statistical test showing that fermented food was superior for inflammation. And the pre-registered primary outcome, the cytokine response score, was not statistically significant in either arm. That is the story this episode is about. The headline-worthy secondary result versus the primary outcome and comparison the paper actually pre-specified. Let us start with the participants and the timeline. This was a small study. 36 people were included in the final analysis, 18 in each arm. They were healthy adults, with an average age of about 52. The full protocol lasted 17 weeks, 3 weeks of baseline, 4 weeks of ramp-up, 6 weeks of maintenance, and a 4-week choice period. The part most people call the 10-week intervention is the ramp plus maintenance period. The fermented food group was asked to reach 6 servings per day. The foods included options such as yogurt, kimchi, sauerkraut, kombucha, kefir, and other fermented products. The high-fiber group was asked to add at least 20 grams of fiber per day. Notice what was not in the design: a no-change control arm. This was a randomized comparison of 2 active diets, not a randomized controlled trial with a passive control. That means both groups were asked to change their diets, and there was no group that continued usual eating for the same period. Now to the result that made the paper famous. The researchers measured 93 serum proteins related to inflammation. In the fermented food arm, 19 decreased under the paper's false discovery rate criteria. The list included IL-6, IL-10, and IL-12B. That is worth reporting. But there are 2 statistical guardrails here. First, those were within-arm changes from baseline. A decrease in one arm and no significant decrease in the other does not automatically establish a difference between arms. To claim superiority, you need the direct comparison. Second, the paper's pre-registered primary outcome was the cytokine response score. That score was not significant in the fermented food arm, and it was not significant in the fiber arm. The primary outcome was null in both. So if a thumbnail says, fermented food beats fiber, ask, which outcome, which test, and compared against what? The study did more than count inflammatory proteins, and this is where the fiber group becomes important. The fiber participants did not respond as one uniform block. They separated into 3 response clusters. One cluster showed increased inflammatory signaling. 2 clusters showed decreases in the same broad signaling families. The low inflammation cluster, with the clearest baseline contrast, began with significantly higher microbiome diversity than the high inflammation cluster, with a reported p-value of 0.037. That does not prove baseline diversity causes a better fiber response. It suggests that the starting microbiome may matter, and it supports the author's more careful conclusion. Responses to fiber were heterogeneous. The fiber arm also showed real biological changes that disappear in simplified retellings. 11 carbohydrate-active enzymes increased and none decreased. Those enzymes mapped to the breakdown of plant cell wall carbohydrates. Microbial protein density per gram of stool increased in the fiber arm and not in the fermented food arm. Neither arm showed an increase in butyrate in this study. So this paper is not evidence that fermented food raised butyrate either. There are limitations beyond the missing control group. The sample was small. The participants were healthy. The intervention was short. And the authors explicitly state that causality was not established. There are also conflicts of interest to disclose. Wastyk and William Van Treuren were founders and shareholders of Interface Biosciences. Justin Sonnenberg was a founder, shareholder, and scientific advisory board member of January AI and Novome Biotechnologies. Those disclosures do not erase the findings. They give the audience context for weighing them. Put that context on screen when naming the lab. So what survives the caveats? Fermented foods produced an interesting within-arm pattern. 19 of 93 inflammatory proteins decreased after the intervention, and fermented food participants showed increased microbiome diversity. Fiber also changed the biology, including carbohydrate-active enzymes and microbial protein density, and people in the fiber arm responded differently depending on their starting profile. What the paper does not show is that fermented food beat fiber for inflammation. The primary cytokine response score was null in both arms. There was no change control group, and there was no direct between-arm inflammation test establishing superiority. This is not a reason to stop eating fermented food, and it is not a reason to dismiss fiber. It is a reason to read nutrition headlines like a scientist. Identify the participants, the dose, the duration, the comparator, and the statistical test. A result can be real and still be narrower than the headline built around it. That is not bad news. That is how evidence becomes useful. Wastyk, H.C. et al. Gut microbiota-targeted diets modulate human immune status. Cell, 2021. PMCID PMC 902079. HTTPS pmc.cbi.delim.nih.gov. Articles PMC 902079. FE50 Trial Registration NCT 03275662. HTTPS clinicaltrials.gov. Study NCT 03275662.